Today let's talk a little bit about protein structure and function, and how the amino acids in a protein determine its shape, and its shape determines its function. We have some learning outcomes today, 3 of them. First, we want to talk about proteins as they're composed of amino acids, and how the properties of that amino acid cause the protein to fold in different ways, and the nature of that folding determines its shape. And proteins can sometimes change shape. As they change shape, they'll change function. So we can start with the discussion of amino acids. Now, I don't want to dwell on all the chemicals, all the bonds. The only reason I've written the structure up here is so we can look at different components of the amino acids, and they'll be color-coded. Every amino acid contains what's written in white here: an amino group, a carboxyl group, a carbon, which is attached to an R group. So everything except the R group is common to every amino acid, without looking at the details of that particular molecule. A different amino acid will have precisely the same components, but a different R group. And that R group can have different properties. That R group might be as simple as a single hydrogen atom. It could be a carbon with 3 hydrogens, or it can be multiple carbons. And the R group can be polar, it can be charged, or it can be hydrophobic. So what determines the property of any given amino acid is the chemical properties of the R group, not the chemical properties of the rest of the molecule. These amino acids then can be joined by a peptide bond. And the peptide bond is shown in the blue rectangle. There's a water molecule that takes off here. We don't care about those details. But we do care that the C-terminal end with all the oxygens of the first amino acid is joined to the nitrogen of the second amino acid. And that bond always looks the same. Now, because the bond always looks the same, there are certain properties of every protein that have to be considered when we consider its shape. So let's look now at the shapes of proteins. This is the same diagram we had before of 2 amino acids joined by a peptide bond. And you'll notice that there are hydrogens and oxygens that are common to every peptide bond. The hydrogens will be partially positive and the oxygens partially negative. So they're going to want to participate in hydrogen bonds, just like we've seen when we talked about water. Proteins can fold in 2 different ways in which the hydrogen that's bound to the nitrogen can be next to one of the oxygens on a different amino acid in the protein. And those 2 types of structures are called a protein's secondary structure. The names are an alpha helix and a beta pleated sheet. Again, I don't want to dwell on those structures specifically, but understanding that proteins need to fold such that these oxygen and hydrogen atoms participate in hydrogen bonds is important. Okay, so if the secondary structure takes care of the components of the peptide bond that are the same for every amino acid, what about the parts that are different? If we look now at the R groups, a hydrophilic R group may be exposed to solvent. It can form hydrogen bonds with the water molecules, or it can be somewhere in the protein where the charged or polar R group can be stabilized by a partial charge or another charge somewhere else. On the other hand, if there's a hydrophobic R group, the hydrophobic side chain is going to need to be buried in another hydrophobic part of the protein. And so the interior of a protein may be largely hydrophobic and consist of the hydrophobic R groups. The way a protein folds in order to arrange the side chains in an appropriate spot is called a protein's tertiary structure. And then we'll see a lot of cases where one polypeptide chain joins with another or is associated with another, and that would be a protein's quaternary structure. So 2 or more different polypeptide chains that are not covalently bound together but interact with each other. With that in mind, we can go ahead and talk about how the shape of a different protein would be related to its function. And to start, I think I'd like to use a protein called albumin. Albumin is one of the major proteins in plasma that gives plasma its properties. And albumin is great for carrying lipids, fatty acids. Keep in mind that fatty acids aren't going to dissolve in an aqueous solution. They're not going to like being around water. So even though fatty acids need to be delivered to every cell in the body, they won't be dissolved. They need a carrier protein. Like all soluble proteins, the exterior of albumin is hydrophilic and likes to be in contact with water. On the other hand, the interior is hydrophobic. So if there's a fatty acid, the fatty acid can bind to albumin so that it's on the interior of the protein and removed from the aqueous solution surrounding it. And because albumin can be delivered throughout the plasma, the fatty acids can also travel through the plasma bound to albumin. Hopefully that makes sense. And then we understand that the interactions of all the atoms in a molecule help determine its shape. So maybe it won't come as a surprise that a protein can change shape when the bonds around it change. And let's look at the case of the insulin receptor. In this particular diagram, the cell is in blue with a black nucleus. The insulin receptor is white, and the insulin molecule delivered in the plasma is green. So on a particular cell, there would be many insulin receptors, so this isn't to scale, but the insulin receptor does cross a cell's plasma membrane, and insulin remains outside the cell. When insulin binds to the insulin receptor changes shape. So the interaction between insulin and the insulin receptor is not covalent, but the amino acids within the insulin receptor, when they're associated with insulin, shift their affinities, and that changes the shape of the entire protein and then changes function. Again, insulin binding, change of shape, change of function. Now what's probably not intuitive is the insulin receptor is an enzyme. It turns on signals within the cell. So when insulin binds to its receptor, the receptor turns on and catalyzes chemical reactions within the cell. And those chemical reactions affect the behavior of the cell. Specifically, glucose transporters are inserted into a cell's plasma membrane. Glycolysis is sped up, the synthesis of glycogen, and the storage of energy. So not only do the amino acids that make up a protein help determine its structure, the environment around a protein helps determine its structure. And if the environment changes, so might the shape. And if the shape of a protein changes, so will the function. So let's summarize what I hope I've covered. Proteins are made of amino acids, and each amino acid has different chemical properties. Proteins are folded, and they're folded so that the components of a peptide bond are stabilized, and so that the differing R chains are in appropriate positions so that everything is happy. Proteins may change shape as their environment changes, so that the interactions between the peptide bonds and side chains remain favorable. And if a protein changes shape, it will also change function.